First, we will briefly go over the RNAscope technology, and then we will provide a detailed overview of the 3 standard generic methodologies that can be used to analyze RNAscope data. Examples will be shown based on different possible staining scenarios, since the ultimate readout from an RNAscope assay is a stain section or slide that will depict a certain staining scenario. We will go over 6 singleplex scenarios and 5 duplex or multiplex scenarios. First, I briefly want to touch upon these 3 points. When running the RNAscope assay, we always recommend to include a positive and negative control to assess tissue and RNA quality. Secondly, the image analyses we show here are done by the Halo software from Indica Labs that we use in-house. However, it can also be done with multiple other software platforms, including ImageJ. Thirdly, the semi-quantitative and quantitative methods provided are guidelines, and it should be noted that there is no universal method that will work for every result or every question a researcher might have. Ultimately, the researcher has to decide what will be the best analysis to answer his or her research question. Also, the quantitation is not always the end goal. The strength of RNAscope is that you get a stained slide or section, and that image itself is of great value because of the morphological context that it contains. Here is a brief overview of the RNAscope technology. It is a slide-based assay intended for the same types of samples that you would use for either IHC or immunofluorescence. This can be either FFPE, so formalin fixed paraffin embedded, or fresh frozen or fixed frozen tissue sections or cells. The RNAscope methodology involves hybridizing the target-specific oligonucleotide probes to the target RNA. The target probe is a pool of oligos that is designed using established informatics tools to provide an assay which we guarantee will be single target specific. Detection is enabled through an amplification procedure that involves sequential hybridization steps to build up a tree-like structure shown here schematically, and which I will describe in more detail on the following slides. Through this method, we are able to localize a tremendous amount of signal to an individual transcript to the point where we can actually detect and visualize individual transcripts as dots. These punctate dots allow for the subsequent quantification of the detected gene expression. The first key feature of the RNAscope technology is the probe design. We depict the oligonucleotide target-specific probes as Zs to emphasize the fact that they have 2 regions linked by a spacer. The bottom of the Z complements and hybridizes to the target transcript, and the top of the Z is the base for the amplification structure to bind. When 2 Zs hybridize in tandem to target sequence, it creates a unique binding site upon which a preamplifier can bind and the amplification tree can be built. A standard RNAscope probe for a target sequence of about 1,000 bases or more will consist of 20 ZZ pairs pulled together and designed to hybridize to each target along a target region. This allows for a tremendous amount of amplification and signal potential. However, a few Z pairs are needed to bind to the target RNA sequence in order to generate enough sequence for molecule detection. The second key feature of the RNAscope technology is the signal amplification. After hybridization with the target RNA, each Z pair creates a binding site at the upper portion of the Z pair for the signal preamplifier to bind. Each preamplifier can bind multiple amplifiers, and each amplifier can further bind multiple labeled probes, sequentially hybridizing to assemble a branching complex, each double Z binding site. Labeled probes can contain a chromogenic enzyme, such as HRP. HRP generates a visible signal after chromogenic reaction with a substrate, such as, for instance, with DAB, and this is detectable under a standard bright field microscope. The labeled probes can also contain fluorophores that also visualize a signal under a fluorescent microscope. This signal amplification strategy allows the visualization of target RNAs as a single dot, where each dot represents an individual RNA molecule that can be quantified. Lastly, background is suppressed with the RNAscope assay because the signal is dependent on two Z's binding next to each other on the target sequence. If both Z's do not bind, then the preamplifier cannot form a stable hybridization at the top of the Z's, and the amplification tree cannot be built. Consequently, no amplification of non-specific hybridization occurs, resulting in little to no background signal. Combined, the combination of the unique probe design and the amplification system assure a high signal-to-noise ratio. The growth and adoption of RNAscope is best exemplified by the number of peer-reviewed publications. We had our first publication in 2011, and since then there are over 800 peer-reviewed publications using RNAscope in high-tier journals. This is illustrated below by some journal covers from last year. The graph to the right shows how RNAscope is highly relevant across multiple fields of research, with almost 50% of the publications in cancer research, followed by neuroscience and infectious diseases. This slide presents an overview of all the RNAscope assays that are available from ACD. We have the Singleplex chromogenic assay for a single target available for both brown and red in manual and automated Ventana and Leica platforms. RNAscope is also available as a chromogenic duplex assay to visualize the gene expression of two targets simultaneously. To visualize 3 or more targets, the RNAscope multiplex fluorescent assay can be used. Staining scenarios for these assays will be shown throughout this webinar. Here, the typical configuration of an RNAscope experiment is shown. Provided that the target is expressed in the sample and the protocol is followed as recommended, the performance of the RNAscope assay is guaranteed. Because tissue samples can be highly variable, proper sample preparation and the right controls are essential to an RNAscope experiment. Tissue samples must be properly fixed and prepared so that there is good RNA quality in the sample and no background due to poor fixation. ACD provides guidelines on how tissues should be fixed for optimal performance of the RNAscope assay. Here are 3 serial sections from a human lung cancer sample on which we performed the manual red chromogenic assay. On the left is the section probed for our negative control probe, DAB, which is a bacterial gene that is not expressed in animal tissues. As observed in the sample, there should be no signal for DAB. In the middle panel is a serial section from the same sample stained with our positive control probe for the housekeeping gene PPIB. As observed in the sample, you want to see fairly uniform target detection with the positive control probe, indicating good RNA quality. Certain tissues may express the positive control probe at higher or lower levels, but in general you want to see expression throughout the sample. From the results for these control probes, we can conclude that there is good RNA quality throughout the tissue sample and that there is little to no background. So we can be confident that the localized pattern for the target probe of interest will in fact be the correct result. On the right, you can see a section from the same lung cancer sample probed for the immune checkpoint marker programmed death ligand 1 or PD-L1. In this sample, we observed robust punctate dots with the PD-L1 probe, indicating expression of PD-L1 in this tumor sample. For more details on how to get started with the RNAscope assay and troubleshooting, refer you to previously recorded webinars available through our website. Next, we will go over the 3 generic methodologies that can be used to analyze RNAscope results. The first method is a semi-quantitative histological scoring methodology based on ACD scoring criteria. The ACD score is based on the average number of dots per cell, and it can range from 0 to 4. An ACD score of 0 corresponds to no staining or an average of less than 1 dot per 10 cells. An ACD score of 4, for instance, corresponds to an average of more than 15 dots per cell with several clusters. ACD scores are semi-quantitative and assigned based on the qualitative visual assessment of the stained slide. Representative images of the 5 possible scores are shown on the right-hand side. The second methodology is an image-based quantitative software analysis. The software will segment the cells and count the number of dots within the cells. The readout of the analysis can be the average number of dots per cell, or the total number of dots in the region of interest, or in the whole section, or it can also be the % positive cells. In this example, for MIC-A and MIC-B expression, the cells are segmented by the HALO software and the number of dots are quantified. The graphical presentation shows the average number of dots per cell for MIC-A and MIC-B. As mentioned, the quantitative analysis and output can be presented in multiple ways, and it is up to the researcher to select how to present his or her quantitative data to best address results. Some examples using this methodology will be touched upon further along in this webinar. The last method uses an H-score and is particularly useful when there is a dynamic range of expression with some cells that are highly expressing the target, while other cells have a very low to no expression for the target of interest. This dynamic range of expression can be quantified for the entire tissue section or for selected regions of interest by binning cells with different levels of expression into separate bins. There are a total of 5 bins ranging from 0 to 4, and each bin is characterized by a certain expression level or number of dots according to the ACD scoring criteria. Here you can see a quantitative image-based example where the number of dots are counted within each cell after segmentation by HALO software. Subsequently, the % of cells that fall into each bin is calculated. The table shows that for this example, 46% of cells fall into bin 1 with an average expression level of 1 to 3 dots per cell, and only 2% of the cells fall into bin 4. This distribution can be visualized in a histogram, as you can also see below. All this data can also be collapsed into one value. The H-score is a weighted formula that will give you one value for the whole section or region of interest, while taking into account the % of cells in each bin. The H-score is derived by multiplying each bin number with the % of cells in that particular bin, and subsequently summarizing all these multiplications across the 5 bins. So for the 46% of cells that fall into bin 1, 46 is multiplied by 1. The overall H-score can range between 0 and 400, and in this specific example, the H-score is 147. Next, we will discuss the data analysis for different staining scenarios that could result from the Singleplex RNAscope assay. And these are the 6 Singleplex staining scenarios that we will go over. The first staining scenario is the homogeneous target expression in a particular cell type. In this scenario, cells display a relatively uniform expression level for the target RNA, indicating homogeneous gene expression among that cell type. The overall homogeneous target expression level of the entire cell population throughout the tissue section can be assessed by measuring the average number of dots per cell. In these example images for human ovarian cancer tissue, you can see the homogeneous expression for MIC-A on the left and MIC-B on the right, with MIC-B displaying a higher expression level than MIC-A. The homogeneous expression level can be analyzed by methodology 1, which is the semi-quantitative ACD scoring method, and methodology 2, which is the image-based quantitative software analysis. When applying the semi-quantitative analysis guidelines, the visually assigned ACD score for MIC-A expression will be 1, and for MIC-B expression will be 2. The expression level can also be analyzed by the quantitative image-based software methodology. For this example, we quantified the average number of dots per cell using the HALO software, but other software can also be used. In the graphical representation, you can see that the average number of dots per cell for MIC-A is 2.9, and for MIC-B is 9.2. The second staining scenario is the heterogeneous target expression in a particular cell type. In this scenario, cells display a range of different expression levels for the same target RNA throughout the tissue section, and this indicates a heterogeneous expression among that cell type. In this scenario, both the expression level and the percent of cells expressing the target at different levels could be of importance. In this non-small cell lung cancer example, the tumor cells in different areas of the section are expressing PD-L1 to a different extent, with medium PD-L1 expression at the left, high PD-L1 expression in the middle, and low to no PD-L1 expression to the right. To assess the heterogeneous PD-L1 expression, methodologies 1, 2, and 3 can be used for the entire section or for particular regions of interest. Semi-quantitative methodology 1 would offer a general visual scoring. The quantitative methodology 2 could be applied to the entire section or to some specific tumor areas of interest in order to derive the number of dots per cell or area. You could also visualize the dynamic expression by using methodology 3 to bin cells with similar expression levels and generate an H-score. A histogram with the percent of cells within each expression category or bin could offer an overall visual representation of the heterogeneous PD-L1 expression profile throughout this non-small cell lung cancer tissue sample. In this third singleplex staining scenario, the target is expressed in 2 or more different cell types, and both the expression level and the percent of cells expressing the target could be important. In this example, mouse colon is stained for beta-catenin, and we can clearly distinguish 3 different cell types that express the target. Higher magnifications for these 3 cell type populations are shown on the right. Panel A visualizes beta-catenin expression in smooth muscle cells, Panel B visualizes the target in epithelial cells, and Panel C visualizes beta-catenin in inflammatory cells. Regarding the data analysis guidelines, a semi-quantitative score could be given to each of these regions representing a specific cell type. Smooth muscle cells would be a score of about 2, epithelial cells 4, and inflammatory cells could have a score of 3. You could also do a quantitative analysis and separate out these regions with different cell populations and calculate the average number of dots per cell in those regions. An H-score could also be generated, although that an H-score for the entire section might not be that informative because of all the different cell types, and rather software should be used to assign an H-score to a specific region or cell type of interest. This is an example of a chart with data generated from 3 samples for which semi-quantitative scoring was done for 1 marker that was expressed in 2 cell populations, namely cell type 1 and 2. Related to the previous image for beta-catenin expression in mouse colon, this could for instance be the smooth muscle cell, the cell type 1, and epithelial cells as cell type 2. For each sample represented in the rows, the semi-quantitative score was generated for cell types 1 and 2 and is shown in columns 2 and 4. Columns 3 and 5 represent the percentage of cells from cell type 1 and 2 that are positive for the target expression. The adjacent chart of percent cells positive in each of the 2 populations for each of the 3 samples allows for easier visualization of this type of data. In this 4th singleplex scenario, the target is expressed in a particular subpopulation or region, and both the expression level and the percent of cells expressing the target at different levels is important. In this example, mouse colon is shown with the LGR5 positive intestinal stem cell population localized at the base of the crypt. Analysis of the entire colon would generate a low average number of dots per cell, and therefore it could be more relevant to hone in on the crypt. And even within this particular crypt cell population, researchers in intestinal stem cell research are interested in the expression levels in the specific subpopulations of cells at position +1, +2, and +3. So it is of particular interest to analyze these specific subpopulations and determine, for example, the average number of dots per cell for these +1 cell populations. If desired, an H-score could also be derived by software analysis. Furthermore, the percentage positive cells can be assessed based on the number of cells with 1 or more dots per cell. This is also a data example that could be obtained with image analyses, methodologies 2 and 3. For one sample with sample ID number 1, there are 3 regions of interest, and for each of these 3 regions, there are multiple readouts, including the average number of dots per cell, the percentage of cells that fall within each of the 5 bins as a measurement of the dynamic range of expression, the H-score, and also the total percent of cells positive for the marker. For a patient tumor sample with 2 regions of interest, for instance, region 1 could refer to tumor cells and region 2 could refer to the stroma. The next category is subcellular localization of target expression. One of the strengths of the RNAscope assay is that it allows for the subcellular localization of expression to subcellular compartments, for instance, the nucleus and the cytoplasm. This could be of particular importance for long non-coding RNAs, where the knowledge of their subcellular localization patterns can provide fundamental insights into their biology and fosters hypotheses for potential molecular roles. Unlike mRNAs, which produce proteins, long non-coding RNAs themselves must localize to their particular site of action, making their location within the cell very important. The 2 long non-coding RNA examples shown here are GAS5 in breast cancer on the left and MELAT1 in lung cancer on the right. For GAS5 expression, dots are present throughout the cells in both the nucleus and the cytoplasm, as indicated by the arrow and the arrowhead. In contrast, MELAT1 expression in lung cancer is predominantly present in the nucleus. For these examples, the actual in situ visualization of the subcellular expression is particularly useful, and a quantitative data analysis could be more challenging because the tissues are actually 3-dimensional structures, whereas the images are 2-dimensional representations. For example, for some cells, the cytoplasm could be present in the section plane, while for other cells, only the nucleus could be included. And this could make it a bit challenging to truly quantify nuclear versus cytoplasmic expression. Nevertheless, the analysis of the expression within each compartment could be achieved with methodologies 1 and 2. Furthermore, percentage positive cells can also be assessed based on the number of cells with 1 or more dots per cell. In this final category for Singleplex staining scenarios, a qualitative assessment of the target expression is needed, and a simple yes or no answer for marker presence is more important than actually quantifying the expression of that marker. The example shows the detection of HPV infection in human samples. The specific probe detects 18 high-risk subtypes of the HPV E6/E7 gene. The condyloma tissue sample on the left is negative for high-risk HPV, whereas the head and neck tumor sample on the right is positive for high-risk HPV. For these types of scenarios, the marker stain slide needs to be reviewed in conjunction with the positive and negative control slides for assessment of marker expression in cells of interest. If the positive and negative control slides have stained appropriately, and depending on the threshold previously set to determine positivity for the particular marker, the cells of interest could be called positive or negative for that marker. Next, we will discuss the data analysis guidelines for the duplex and multiplex scenarios with the expression of 2 or more targets respectively. Here you can see an overview of the 5 different duplex and multiplex RNAscope staining scenarios that we will go over. The first duplex scenario is the expression of 2 independent targets in different cell types. In this scenario, the expression patterns of 2 genes of interest are simultaneously detected, and both the expression level and the percent of cells expression each of these 2 targets could be of importance. In this mouse brain example, the 2 targets of interest are DRD1 in red and DRD2 shown in green. Each of these genes marks a different subset of medium spiny neuronal cells in the striatum. Essentially, what one could do to determine the target expression for each of the 2 genes within a particular cell type is use methodology 1 and give a semi-quantitative ACD score to the target expression in each of these populations. For instance, a score 4 for DRD1 and also for DRD2. One could also use methodology 2 and quantitate the average number of dots per cell for target 1 being DRD1 and target 2 being DRD2 using image-based software analysis, or determine how many cells are positive for DRD1 and how many cells are positive for DRD2. An individual H-score could also be derived for each of these two targets. This is an example of the halo analysis of a non-small cell lung cancer tissue section stained for CD45 in red and PD-L1 in green with the RNAscope Duplex assay. The chromogenic duplex image for both targets is shown on the left. The two middle panels show the probe markup by halo for the green and red probe. After cell segmentation, the degree of expression for each of these targets is visualized by the color shading of the cells. Dark green and red indicate cells with high PD-L1 and CD45 expression respectively. On the right-hand side is the dual probe markup, with green and red indicating cells with either PD-L1 or CD45 expression respectively, while yellow highlights cells with co-expression of both these targets. The formula shown here represents different ways of calculating the co-expression of two targets, being target A and B. The first formula calculates the number of cells that express both target A and target B out of the total number of cells. The other two formulas calculate how many cells that are expressing target A also co-express target B, and reversely, how many cells that express target B also co-express target A. This example output chart builds upon the earlier chart for one target, but now for each of the 5 samples, there are 2 targets, target A and B. For each target, average dots per cell, the percentage of cells that fall into each bin, the total percentage of cells positive for the target, and the H-score are generated. Additionally, the percentage of cells that are dual positive and that co-express both markers and the percentage of cells that are dual negative are generated. In this chart, the percent dual positive refers to the first formula, in that it is the percentage of cells that express target A and target B out of the entire population of cells analyzed. However, based on the research question, this column could also be adjusted to reflect the formulas shown in number 2. So there are different readouts that the researcher could use to quantify co-expression, and it ultimately comes down to the biology and the research question or hypothesis that needs to be answered. In the second duplex scenario, cells co-express 2 genes simultaneously in the same cell. This allows for the detection of a target of interest along with a cell type marker for the cell type that expresses that target of interest, or the co-expression of 2 targets within the same cell. In this example, we show the co-expression analysis of the immune checkpoint markers TIM3 in red and PD-L1 in green in non-small cell lung cancer. On the right, the halo output from the image analysis is shown for a tumor compartment. The cells singly expressing one of the markers are shaded in red for TIM3 and in green for PD-L1. The cells that are co-expressing both TIM3 and PD-L1 are shaded in yellow. The data could be presented as the cells individually expressing TIM3 and PD-L1, or as the total number of cells co-expressing TIM3 and PD-L1, or the number of TIM3 expressing cells that also co-express PD-L1, or reversely, the number of PD-L1 expressing cells that also co-express TIM3. The third type of scenario is the target expression in a rare cell type. Essentially, very few cells are expressing the target of interest in this scenario, and therefore identifying the number of cells that express the target of interest could be more relevant than the average target expression level per cell. In this lung cancer example, a small number of cells is expressing CD8 in red And these CD8 positive T cells are infiltrating the PD-L1 positive tumor area with PD-L1 expression in green. One can qualitatively and quantitatively assess the expression pattern by using methodologies 1 and 2 for each of the targets. However, in the case when very few cells are expressing the target, the average expression level might not necessarily be the most useful readout, but the number or percentage of, in this case, CD8 positive cells in the region of interest could be more informative. The fourth scenario is about cell population interactions. Here, the researcher is interested in specific cell populations and their spatial relationship to one another. One can assess the number of cells expressing the targets of interest and their proximity to one another within the tissue environment. In this lung cancer example, the CD45 positive cell population is shown in red, and the PD-L1 positive cell population is shown in green. Regarding the spatial interaction, one can see how the CD45 positive stromal cells are infiltrating in the PD-L1 positive tumor area. For quantification, methodologies 1 and 2 can be used. However, it should also be noted that valuable interaction-related information is provided by the visual image itself. Lastly, we have the multiplex target expression analysis. In this scenario, multiple target expression patterns in one or multiple cell types are investigated. There is a tech note available online that provides very detailed guidelines for how to approach multiplex fluorescence data analysis. What is shown here are a couple of examples from different research areas. This first example is human breast cancer that shows the multiplex expression of keratin 19 in green, PKM1 in red, and MKI67 in white, with keratin 19 being used as to detect tumor cells, PKM1 as an endothelial cell marker, and Ki67 as a cell proliferation marker. So basically, this image shows 3 different targets marking 3 different cell types or populations, and the researcher can use any of the 3 methodologies to analyze the data. This is another example, but from the neuroscience field, where the multiplex expression is shown for VGLUT1 in red, VGLUT2 in green, and VGAT in white, and they all mark different excitatory and inhibitory neuronal populations in the mouse brain. This data chart generated for the multiplex data analysis has a similar format than the singleplex and duplex data, but now data is generated for a third marker being target C. For each of the 3 targets, you can see the average dots per cell, the percentage of cells that fall into each bin with a certain expression level, the total percentage of cells positive for the target, and the H-score. Also, because of space limitations here, the percentage of cells that fall into each bin has been taken out, but would certainly be generated for each target. Also, the percentage of cells that co-express A and B, B and C, and A and C, and all of the 3 targets together are generated. And also of note is that these percentages are expressed out of the total number of cells in the area analyzed. However, again, based on the research question, these columns could be adjusted to reflect, for instance, the number of cells positive for target A that are also co-expressing target C and target B. And again, ultimately, it comes down to the biology and what a research question or hypothesis needs to be answered. To summarize, we discussed 3 generic RNAscope data analysis methodologies that can be used across multiple research applications to answer different types of research questions. And we provided about examples for 11 staining scenarios. If you would have any questions, you can always email or call support. Also, available resources can be viewed on our website, and support is available in the US, Europe, and Asia Pacific. Our website also offers plenty of information from technology to support to products, applications, etc. Everybody who has also registered for this talk will get an email with the option to download the data analysis guide once the document is finalized. So what is listed here are some frequently asked questions. So one of the questions is, why should I use positive or negative control probes? So we always recommend using both positive and negative control probes, as samples may not be prepared according to the sample preparation guidelines that we provide. And the RNA quality is also not always known. The species-specific positive control probe, PPIB, will help to determine whether the RNA in the tissue specimen is of sufficient quality for detecting the RNA biomarker of interest. And the negative control probe, which is the bacterial gene DAP-B, will help to determine whether the tissue specimen is appropriately prepared. So that's why we use these two control probes and why we always advise to include them in the experiment. The second question that comes up is, what does each dot represent? Is it a single RNA strand or a collection of RNA signals? Well, for the RNAscope assay, standard RNAscope probes consist of 20 ZZ probe pairs, and they target a region of about 1,000 unique bases. One dot represents one single RNA transcript. And in cases where the copy number of transcript is, for instance, very high, it is possible that signals can appear as clusters. Now, does the intensity of the signals matter with RNAscope is another common question. Well, the signal intensity is not relevant to expression levels. Differences in signal appear only at smaller or larger dots, and this reflects assay conditions, sample preparation, and the number of probe pairs that are actually binding. Dot size does not reflect the expression level, and in some cases where the copy number of the transcript is higher, the signals will appear as clusters. So, if there are any more questions. So one of the questions that came up during the webinar is whether RNAscope readouts need to be normalized to actually compare samples. Well, RNAscope readouts do not need to be normalized in order to compare expression levels across samples, as long as the tissues were similarly processed. And this relates to their storage, for instance, to how they are fixed, and also the thickness of the sections. What is also important is that all samples pass quality control, which we talked about before, being the positive and negative control probes. In that case, when all of this is okay, RNAscope will offer absolute readouts or numbers, and these represent the number of transcripts. Okay, so another question that just came up is whether target expression that appears as clusters, whether that's possible, and whether it will interfere with data analysis. Well, in cases where the copy number of the transcript is high, these signals can appear as clusters. Well, what is important is that for the quantitative analysis of the number of dots by the HALO software, the parameter can be set to determine the dot size and diameter for detection. So they can be set up front. And as such, clusters will be broken down into the number of dots with the selected settings that can fit into that cluster. This is not necessarily the same for other software, but this is how HALO software deals with clusters and getting a dot count out of them. Also related to the HALO software is people ask where they can access the software. Well, this software is available through Indica Labs, but also here in ACD we offer the service for HALO data analysis. So there's a lot of questions actually coming up, and support at this point is chatting and answering them. And some more that are coming up are related to the magnification. So people ask, what is the magnification of the slides that is used to analyze the data? Well, the answer to that is that we use 20x images to analyze the data. Another question that came up is whether the Indica software is also available for purchase. So as mentioned before, yes, it is through Indica Labs, and we offer a service. Indica Labs can be contacted directly at info@indicalabs.com. Another question, a technical question related to the RNAscope assay that has come up is what is the recommended thickness of FFPE samples? Well, the answer to that is that the thickness of the sections that is used for the assay lies between 4 and 6 micrometers. So, okay, this is a bunch of the questions that have come up during the webinar. Please don't hesitate to contact us with any more questions. And if answers to your questions were not provided at this point during the webinar, we will reach out to you and follow up with you after the webinar. Thank you for attending. So I will just note that the chat will be open for 5 more minutes to get direct answers, and as mentioned, otherwise we will get back to you later. Thank you for attending.